and welcome to the MAPS Canada webinar series, Examining the Psychedelic Renaissance, Season 2. My name is Reverend Shannon Smedilla, and I'm your host for the series and a longtime volunteer of MAPS Canada. We are beyond excited to bring to you Season 2 as we've had an extremely potent and successful Season 1, full of amazing speakers, researchers, and of course, attendees like all of you. And it's so great seeing some familiar names pop up in the chat today. We are now in episode 5 of Season 2 of our web series, and we've got some amazing speakers on board. Each of these speakers and panels will be showing up to share important information and their take on the psychedelic renaissance. At MAPS Canada, the Multidisciplinary Association for Psychedelic Studies, we are on the forefront of conducting studies to develop psychedelics into safe and legal medical treatment since our formation. To conduct in public scientific research and education supporting the beneficial use of psychedelic medicines, MAPS Canada relies on public donations and fundraising initiatives. The funds raised from this webinar series will go to fund this research, so thank you for being here. There are a few different studies that MAPS is currently working on, and you can see full details at mapscanada.org. So some housekeeping items for today. We're going to start out with an amazing conversation with Dr. Joe Tafur. He's our special guest. After that, we will start our Q&A session, and we'll also welcome a couple of his co-investigators on his study onto Crowdcast for that. So make sure and put your questions in the Ask a Question, and also let us know where you're tuning in from in the chat. So we see people from all over the world today. We have Arizona, Gibsons. We have someone on the ferry. We also have Victoria, BC. We have Mexico, Vancouver, Winnipeg, Burnaby, San Diego. More from Vancouver, New York City, Santa Monica. We have Ontario. We have Phoenix. We have Waterloo. We have North Carolina, Santa Cruz, Colorado. And the list goes on and on and on. False Creek there. And we have London, Ottawa, Nova Scotia. Hey, representing Canada East. So we're about to get started. And before we do, let's talk a little bit about Dr. Joe Tafur. Joe Tafur is an integrative family physician, educator, ayahuasca curandero trained in the Peruvian Shipibo lineage. Dr. Tafur completed his medical studies at the UCSD School of Medicine and his family medicine residency at UCLA. He also completed a 2-year postdoctoral research fellowship at the UCSD Department of Psychiatry. He is the author of The Fellowship of the River, a medical doctor's exploration into traditional Amazonian plant medicine, forward by Dr. Gabor Mate, in which he shares his fascinating journey into spiritual healing work. He's the founding member and medicine carrier of the Church of the Eagle and Condor, a multicultural spiritual community based in Arizona. Dr. Tafur also co-founded the traditional healing center, Niue Ro, Centro Espiritual in Iquitos, Peru, where he was trained over 7 years in Shipibo curanderismo, traditional spiritual healing involving ayahuasca ceremony and Amazonian master plants, sometimes referred to as shamanism. He continues his medical work through the Ocotillo Center for Integrative Medicine in Phoenix, Arizona. Dr. Tafur also founded the nonprofit organization Modern Spirit, dedicated to demonstrating the value of spiritual healing in modern healthcare. Modern Spirit is currently funding the Modern Spirit Epigenetics project in collaboration with USC and MAPS, which is investigating the epigenetic impact of transformative healing experiences observed in the MAPS MDMA-assisted psychotherapy for PTSD trial. This fall, Dr. Tafur will be reaching his live online 8-week river course, an integrative approach to psychedelic-assisted therapies and sacred plant medicine. And that is actually now sold out. So today we're going to be talking about epigenetics and psychedelic-assisted therapies, spiritual healing in modern healthcare. Much needed. For the last 2 years, Modern Spirit, a nonprofit dedicated to demonstrating the value of spiritual healing in modern healthcare, has been collaborating with MAPS and the USC Department of Psychiatry. Participating study centers have been collecting saliva samples before and after the MDMA-assisted psychotherapy intervention for PTSD. The initial set of samples are now being analyzed at TGen in Phoenix, Arizona. These samples are being evaluated for epigenetics changes, looking for ways in which the therapeutic intervention, which has led to so many transformational experiences, may be influencing the way genes are expressed in study participants. PTSD has been associated with certain epigenetic modifications. Lasting healing from PTSD may similarly involve change at the epigenetic level. Dr. Tafur will discuss this groundbreaking study, Modern Spirit Epigenetics Project, its formation and current progress, as we explore where the intersection between biology, emotion, and spirituality. You can learn more at modernspirit.org, and there will be a link in the chat. And I believe they are also fundraising, and there will be a link in the chat for that as well. So without any further ado, let us welcome Dr. Joe Tafur. Welcome, Joe.
Thank you. Thank you very much. Nice to be here. Thank you to MAPS. Thank you to everybody who's participating. And yeah, here we are on this call. Screen time.
It's so great to have you here, Joe. So I'm going to turn the screen over to you, for your presentation, and then when you're done, we'll invite your co-investigators on as well. So Thank you again so much.
Thank you. So yeah, my goal is to run through some material about, like I said, the history, or she said, the history of the study and the rationale for the study and the update on the study, and then try to get the co-investigators on for questions. So I don't want to drag out the beginning part, but I'm going to get some information out. We'll see how it goes if I overdo it again. But just to go to the slides for a second, let's try it. Application window. So here's the slides. I believe I'm in the slides. And, the introduction, I'm going to just pull out of the, I'm going to have to, how do you turn it off? How do you turn off the screen share? Stop sharing. Here we go. So just the intro, okay, just to get things going. So I was just hanging out with a friend of mine, close friend, who's involved in psychology and the rollout of a lot of new things. And there's a big movement around ACEs. Perhaps you've heard about ACEs, adverse childhood experiences, and the way those kind of things can traumatize someone, and then stays with them. And it leads to all these healthcare issues down the road. And so now it's becoming part of the interview process of patients to try to ask them about their ACEs. And similarly, we used to have this example of PTSD, where you have this trauma, and then we have this stable shift that happens in the person from that trauma that stays with them and extends into their life. And then the issue is that some people can get better from some of that stuff, and some people have what is considered treatment-resistant trauma. They've got this heavy trauma that's affecting their mental health, it's affecting their physical health, and nothing's really helping them. Let's say, like, you can imagine like a Vietnam vet with this extended history. So needs to be a lot more done to understand that kind of trauma, and something to help bring more hope to that kind of trauma. That for me as a doctor, in my initial training, things like intense combat trauma or intense sexual abuse were the kind of things that people weren't really sure, that we could help. So now epigenetics is opening a pathway to that understanding and hope. So we talked a little bit about epigenetics. So this idea that the biology of this kind of trauma imprinting that happens is perhaps epigenetic. I'll try to explain that a little bit more. And that if we could understand more about how that epigenetic problem is linked to their health problem, and that we could maybe see that epigenetics change, just as it was imprinted and changed in response to the trauma, perhaps it could be improved. And so there's these transformative healing experiences that we see, like the MDMA-assisted psychotherapy trial for PTSD that MAPS is running. And they're seeing these humongous shifts, people that have had PTSD for 20 years. And now after a 12-week intervention, at least half to 60% of them don't have PTSD anymore. So it's some kind of transformative healing. And, what does that mean? What does that mean in their biology? So if we can understand what's going on underneath that, we can learn how to help these kind of people more. And we can also get more hope for those people. But furthermore, we also strengthen the case for psychedelic assisted therapies. So why is this important? So that's why it's important. Epigenetics and its role in this kind of healing work for me is extremely important in increasing our understanding and increasing hope and then also strengthening the case for this kind of therapy. So I'm gonna try to go back on the screen share if I can. Just kind of pathetic that I can't. There we go. So I'll probably just stay in the slides to make it easier for a little while. So I'm here to kind of update everybody about this. This is the Modern Spirit Epigenetics Project. So this is what it's called at the university: Epigenetic Regulation of Stress-Related Genes in relation to MDMA-assisted psychotherapy treatment for PTSD. So the principal investigator, it's not me, I'm just the guy who goes out and talks and tries to raise the money and tell people about it. It's Raheel Khan at USC Psychiatry. The main co-investigator, or these are two people that I hope to have on to help us answer questions, is Dr. Candice Lewis, who is a fellow at TGen, Neurogenomics and ASU Psychology. She's actually doing the hands-on, the lab work. So this study is in collaboration with MAPS. Of course, we depend on MAPS for everything. We're piggybacking on the MDMA trial. USC is the home of this study, leading and coordinating the study under Raheel. And then TGen is the location where we're actually running this epigenetic stuff, the lab work. So my background of how I got into all this work is, whether you know or you don't, is in Peru. So my experiences around this kind of transformative healing, or my exposure to psychedelic psychotherapies or psychedelic therapies, was really through sacred plant medicine and my work in Peru for years at Rao. And I'm just going to read for a moment here a little quote from Albert Hoffman, the guy who discovered LSD. They asked him in this book, Higher Wisdom, what can we learn from the so-called primitive cultures who use psychedelic substances as part of their religious practice? He says the most important thing is that they use psychedelics in a spiritual framework, and we don't. We must learn from them, and we must identify the right structures. We must find new uses. I can imagine that it may be possible to create meditation centers for psychedelic use in natural surroundings, where teachers could have experiences trained to become adepts. I perceive this as being possible, but first, psychedelics will have to become available to medicine and psychiatry, and then it should be made available for such spiritual centers. Basically, all that we need to know, we can learn from how the primitive people use psychedelics as sacraments in a spiritual framework. We need such centers, but we also need the psychiatrists. These psychiatrists must become the shamans of our times. So this idea that we can learn from traditional use. So I had all this experience with ayahuasca. And in my book, I talked about an experience that I had in learning curanderismo and singing ícaros and mystical healing songs in ayahuasca ceremony. A lot of it is a visionary work that we're doing on people. And I describe a case in the book where I'm singing to somebody named Lisa in the book who was suffering from migraine headaches. And the ayahuasca presented this vision of the DNA strand wrapped in these protein molecules. And that was like the DNA packaged. And there was this little black dragon floating through the nooks and crannies and the crevices of this DNA, packaged DNA. And I received this message from the ayahuasca that Joe, her problem is not in her genes, it's on her genes. That's what we call epigenetics, that which sits upon the genes. And so from this vision and then watching her subsequent healing through the kind of work we were doing, she had a dramatic shift, transformative healing experience around her migraine headaches. I started becoming very interested in epigenetics. So this is a little thing showing this DNA strand wrapping around the proteins. And in this case, showing how these methyl groups, these little molecules, can be tagged onto the DNA strand. And we know about how as you tag the strand with these methyl groups, the methylation process, it influences the way those genes are expressed, the underlying genes. And this is becoming really interesting and important for all kinds of illnesses. And they're showing how many things can lead to this methylation, the differing methylation. They said that development in utero, childhood, chemicals, drugs, pharmaceuticals, aging, diet, exercise, social interactions, parenting, all these different things. And then that ends up leading to epigenetic shifts that can be connected to disease, like cancer or autoimmune disease or mental health problems. And the thing about this is like a conditioning problem. It's not their genes, it's what's on top of their genes. It's software versus hardware. So this is kind of the focus. So I had this experience, and so I came to MAPS 2013. I got invited to speak. And I presented this slide just based on basically my ayahuasca visions and starting to explore and say, is this real? Is there really epigenetics connected to this stuff? I knew a little bit about it, but I didn't know much. So I presented this slide. I think we should start exploring the possibility that we at the ayahuasca healing centers are cleaning allostatic load, which is basically stress, wear, and tear, pathologically imprinted epigenetics from our emotional centers, which, by the way, are hardwired into our tears and our vomit. So that's what I got to and started getting really interested in trying to see who else was into this. That got me on this road to start looking at more and more scientific papers and, building an argument as I kept working down in Peru and seeing more people go through stuff. And here's a couple papers showing how different maternal rearing, so raising these monkeys differently, was changing the methylone, was changing the way the methylation of their genes, and that you could pick that up not only in their brains, but you could pick it up in T cells in their blood. So this imprint of the trauma could be found in the blood, not just by, dissecting their brains. Here's another paper. They show early life trauma depression in the cortisol receptor gene, the glucocorticoid receptor gene, epigenetic perspective. So this idea that early life trauma would be connected to some kind of epigenetic imprinting, and that could be connected to depression later in life. So then there was other couple studies that caught my eye. This mouse study, when people started talking about inheritable epigenetics, because that was linked to ancestral trauma ideas that we had down in the jungle. This idea that if you condition a mouse to be stressed against a certain smell, that would be passed on for multiple generations, that would be actually part of what instinct is for that mouse and its future generations, but that there would be maladaptive versions of that. So we have Bowers and Rachel Yehuda, trauma researching powerhouse, brought out this stuff, intergenerational transmission of stress in humans, that concentration camp survivors would be carrying epigenetic imprints from their trauma in the war, and this would be passed on to their kids and lead to increased prevalence of anxiety disorders, for example. And it wasn't just because of the parenting, it was actually because of passed on trauma. So all of these ideas came together for me in my book, and I presented my book in 2017 at MAPS. And I said, hey, potential role of epigenetics and ayahuasca shamanism. So I was really focused on the work we were down there doing in Peru and saying, hey, maybe we're affecting epigenetics because I'm seeing these big transformative healings around trauma. And so as I did that research for the book, I started looking for evidence of interventional modification of epigenetics. So not just trauma causing change, but is there any evidence that this software can be changed by treatment? And so there were a few papers at that time about antidepressants, about psychotherapy leading to epigenetic change, and even meditation, so altered states of consciousness. So what about these transformational healing processes? What about these big healings that we saw down in Peru, that we see sometimes that the MAPS trial is reporting on? What about those? That we know trauma is related to those. We know there's epigenetics correlated to those traumas. And then we see this big shift. Is there an emotional biology that's shifting? So the book got me to meet a lot of people. And I met Dr. David Rabin and Dr. Ben Kelmendi, who were, they're like, we agree. We're interested in the same ideas. We think epigenetics are at play in this MDMA-assisted psychotherapy healing and for the treatment-resistant PTSD. And, let's see if we can start a research project around ayahuasca. And we were in Peru and we tried to do that. We started investigating. It's just logistically, it was very difficult to pull that off. And then meanwhile, Dr. Rabin had been connected to the MAPS study. Ben Kelmendi had very many connections with MAPS. And so we said, what if we just try to do piggyback on their study, got saliva swabs, and try to see if we could show epigenetic shift there, where they're seeing the same kind of healing that we're seeing. So we still needed a home for the study. And so I asked my old friend, Raheel Khan, that I went to medical school with, who had come to medical school saying, I'm going to study psychedelics and meditation. And we all thought he was, that was just off the wall. But his thesis work turned out to be with the UCSD Department of Neurosciences, comparing and contrasting the impact of long-term mindfulness meditation practice and the acute effects of the psychedelic drug psilocybin on perception, attention, and brain activity using EEG methods. So now he's assistant professor. All these years later, I went to Peru, and Raheel became assistant professor at USC and director of the Center for Mindfulness Science. So he was over there in Switzerland at the Vollenweider lab doing research on psilocybin. So as he agreed to take on the study, to be the PI, principal investigator, USC would house the study, and Modern Spirit started fundraising. He said, okay, we'll get started. We talked to MAPS. let's start collecting saliva samples was a big issue, because there's a window as MAPS negotiates with the FDA and tries to find out how many people can we do this research on, and when can we do it. So to collect the samples while people are going through the study became really important. So we just had to get the ball rolling. So let's start collecting. We'll figure out analysis later. There's a lot of labs that might be able to do it. And then in 2019, met Dr. Candice Lewis at the Arizona Psychedelics Conference. I myself live in Phoenix. She was presenting on trauma, psychedelics, and epigenetics, and very comprehensive kind of approach to the topic, was very impressive. She also happened to be a Fulbright scholar to Zurich and had been at the Franz Wollenweiter lab. So, they had a lot in common, Raheel and her. She's a fellow at TGen, the Translational Genomics Research Institute, and ASU Psychology. So Candice says, I can do the work. I do this work. I do this epigenetics lab work. So we were ready to take it on. And then later on with David and Ben and MAPS Connections, we met Dr. Rachel Yehuda, who is just a leading trauma researcher, director of the Traumatic Stress Studies Division at the Mount Sinai School of Medicine, and everything you see there. And so she's there also as a co-investigator to advise us and hopefully to help us in the analysis as we move forward. So it's a big team, a beautiful team, strong team. And so what's the update? How far? October 2020, here we've got the pumpkin with the mask, COVID time. So far, we've gotten 23 pre and post saliva samples. So that's what we've got from these different MAPS centers, areas that are running the trial, and they're ready to go. And so we ran them through the lab, and it's done. They're ready to roll. We have another 10 pre's that got interrupted by the COVID, and we don't have the post. And so we're trying to track down those posts through mail, saliva collection, and we're waiting till October 24th. It's our understanding that they're going to unblind the MAPS trial, who was on placebo, who got MDMA, and then we're going to be able to talk about our results. So hopefully this is weeks to a couple months away from really reporting on the study. And then we've also been invited by MAPS to go ahead and try to do with the next round that they're going to open up now in the post-COVID phase 3, next MAPS 2 it's called, more people, more numbers. And so we're going to be doing saliva collections at people's homes. So what is PTSD? Just a quick review for everybody. This is the definition according to the psychological kind of medical understanding: atypical psychological and physiological response that can occur among persons exposed to a potentially traumatic event involving life threat, serious injury, or death. So there is these subsequent criteria that are part of the diagnosis that kind of define, what is PTSD. So when somebody comes into the study, that they meet this criteria, and when somebody is considered to no longer meet criteria for PTSD, they don't. So that they kind of had a healing, I would say. So what is going on with the MAPS trial? So all this revolves around the breakthrough work that MAPS has been doing. So they had their first publication about, or the one that I was aware of, 2011. They started showing, hey, this is effective. This is a really big deal. I think the initial trial showed that people with average duration of 18 years of PTSD, something like that, they had over 60% of their participants no longer meet criteria after a 12-week intervention, and then that sustained, a year later and beyond. So it was a breakthrough in psychiatry and a breakthrough in mental health. They had subsequent studies showing that it's safe, it's not harmful. Everyone that was worried about MDMA and using it, they're using it, maybe 3 times in a 12-week period. And then they even, because of the impressiveness of their work, starting to get more conservative elements involved. Military veterans, firefighters, police officers, also showing, positive results. So they just released this recently. Now this is the kind of summary of that work. MDMA assisted psychotherapy for treatment of PTSD, the review of their phase 2 trials. So it turns out MDMA assisted psychotherapy was efficacious and well tolerated in a large sample of adults with PTSD. This is again, PTSD. So 72 active treatment and 31 control or placebo. So because of the impressive results, kind of the groundbreaking results that this study was making, they have expanded into phase 3 trials and led to FDA granting breakthrough therapy. So this is this idea that we hear about that maybe in a year or 2 years or less, this is going to be FDA approved because the research is so impressive. So what about trauma, PTSD, and epigenetics? What can we say about the science? Can we track methylation patterns around PTSD? Can we see that Is methylation, are methylation patterns linked to this trauma? And is the intervention, is the healing, can we track that? So that if we measured those kind of gene areas around MDMA treatment, would we see results? So we have Rachel Yehuda and Nabir. There's in 2009, the relevance of epigenetics to PTSD. So way back when, in 2009, I hadn't really thought much about this. I hadn't had that vision, I hadn't trained in shamanism, but I did take my first group down to Peru. And among that group was a family friend of mine, Russ, Vietnam vet. And he had a really transformative healing. Well, that same year, before there was a lot of data, Rachel Yehuda and her colleagues had identified that although there are currently no findings that suggest epigenetic modifications that are specific to PTSD or PTSD risk, many recent observations are compatible with epigenetic explanations. These include recent findings of stress-related gene expression in utero, meaning in the womb, contributions to infant biology, the association of PTSD risk with maternal PTSD, and the relevance of childhood adversity to the development of PTSD. Well, this was real interesting to me. Here's my teacher, Ricardo Marino, in the Amazon, Shipibo healer, that his approach with people like Russ and the other people we were working with was all about cleaning the energies of our traumas, our strong experiences, our strong emotional experiences, which tended to be, in utero, infancy, childhood, adolescence, adult life, in our environment, relationships, and even ancestral. And that this was all linked to what epigenetics was saying. Hey, these are all kind of epigenetic areas where trauma could imprint. And so that's something that she had also suggested with her colleagues. So is how you say clean in Shipibo, to clean the energies in the songs. So in the book, I kind of got into this idea of an emotional body. So we could say, okay, this methylation is happening. This DNA is getting marked. But what does that mean for the physiology? What does that mean for emotional biology? What happens to the person when the DNA is methylated, this or what happens when the trauma happens? So this idea of the existence of an emotional body, the psychoneuroendocrine immunologic network was this network that I had learned about in my own little brief time in research, is kind of the mind-body system that has been illuminated by stress research, people trying to understand how does stress affect illness, how does stress lead to high blood pressure, why when you get depressed, you have immune function problems, and why does emotional trauma cause problems with the stress response system, like in PTSD. So you have one of the core elements of this emotional body that I call it, or the PNEI network, is this HPA axis, right? The hypothalamus, pituitary, adrenal glands, where that's controlling cortisol levels, for example, and adrenal production and people's response to stress. So this PNEI system, this emotional body, I'm saying it's a limbic system, the emotional part of the brain that is also connected and networked with parts of the immune system, with parts of the autonomic nervous system and the endocrine system, the hormones, like I was talking about, the pituitary and the adrenal glands. So this whole network, this system that is affected by trauma. So one of the most obvious examples is in PTSD, the way the HPA axis, this stress response system, is affected. So we know there's research to show that people with PTSD have problems with this system. So here's an example. But now we have this epigenetics. So now we say if we have epigenetic modulation, if we tag the glucocorticoid receptor, okay, so now this is the cortisol receptor. This is the receptor that the dexamethasone was acting on Trump, lighting them up, helping them feel wide awake. So cortisol levels, when they peak in the morning, it helps wake you up. And that's glucocorticoid. That's what we call it. The receptors are the ones that respond to that cortisol. So these are modulated epigenetically in response to trauma. We've measured that, and that affects the way cortisol levels, the way they do their little dance through the day. Okay, the slope and the peak and all that stuff, it changes. It doesn't go as high in PTSD, it doesn't wake them up as much, and it stays a little high. It lingers, and the slope is flatter. So this emotional body, what can we say about cortisol receptor methylation and the HPA axis in adolescents, for example? They show, hey, if you look at all these adolescents, they're saying that methylation levels were associated with a flattened cortisol recovery slope, indicating a delayed recovery time. These results suggest that methylation of the NR3C1, which in this case, once again, is the cortisol receptor, may impair negative feedback. So it sounds like a lot of complex science. Basically, the feedback loop of that cycle I just showed you is affected, we know, by trauma, and we know it's affected by the methylation that's been correlated to trauma. Here's one of Dr. Lewis's studies. Harsh parenting predicts novel HPA receptor gene methylation, and cortisol methylation predicts cortisol daily slope. So one more, very scientific slide that's basically showing, something about how epigenetics, our results suggest possible epigenetic mechanisms linking the relationship between harsh parenting and negative health and behavioral outcomes. So this is kind of where we're getting with the science. And it's about how this system, this HPA axis, is affected by trauma, how that affects epigenetic methylation of things like the cortisol receptor gene, and how that affects the feedback of this little cycle that's going on here. So PTSD, in my estimation, the way I talk about in the book, is about this emotional body where there's not just the cortisol receptor, but there's a lot of different receptors involved that we're starting to gain knowledge about as far as epigenetic modulation in response to trauma. So there's areas of the nervous system, there's this BDNF, there's other serotonin transporter, all these different areas, cortisol receptor we already mentioned, and then we see immune function change. So it's this wide change across this network and a lot of different gene clusters that we could track. So if we're going to study this, we were interested in studying not just cortisol receptor, but a wider range of gene clusters to try to capture the kind of diffuse things that are happening with the emotional body. Here's another study kind of to indicate a little bit about this complexity of what we're learning. Here's Dr. Lewis again. Epigenetic differences in inflammation genes of monozygotic twins are related to parent-child emotional availability and health. So you have these twins, and then at these different times, these different windows of development, you can show that we know they have the same DNA, but then if they are exposed to different emotional availability, that's going to change methylation, and that's going to change the way their immune system functions down the road. So suggesting the methylation of immune genes may have functional consequences for general health. Here's a further example of that in PTSD. So epigenetic and immune function profiles associated with PTSD. So you got PTSD, and they show these methylation profiles that are linked to the trauma, and it results in changes in the gene expression associated with altered immune function. So now these people are more immune compromised. They start coming down with this disease CMV, which only affects people who are immune compromised. So the immune compromise that they're suffering from, this CMV that they have, is actually trauma related. And if we could heal this trauma, they wouldn't be perhaps so vulnerable to that illness. So that's kind of trauma, PTSD, and epigenetics really fast and firing at everybody. But I just want to kind of cover some of the rationale and help people understand what we're trying to do. So this network, as I mentioned, there's different parts of the body that have tissue-specific methylation patterns that are linked, as I mentioned earlier. So we get an idea of what's going on in the brain because certain cells in the body also are methylated in similar patterns. And in this case, the buccal cells of the mouth, these oral mucosal cells, you can swab them and you get an idea of what's going on in the brain. And so what about therapeutic modification of epigenetics? So we've got this dragon swimming over the genes and the proteins, and we want to heal it, Down in Peru, Shipibo style, I'm going to try to sing it out. I'm going to try to sing it out and help this person that way. What kind of research has been shown about can this software be shifted? So I mentioned the antidepressant work, the meditation work, the psychotherapeutic research that all showed, yes, it is possible. Here's a Dr. Yehuda again, showing, moreover, psychotherapy constitutes a form of environmental regulation that may alter epigenetic state. Finally, the results further suggest that different genes may be associated with prognosis and symptom state respectively. This idea that the results support research indicating that some, again, cortisol-related genes are subject to environmental regulation throughout life. So this software, even in PTSD, can change and can change within the lifespan. So now we get into this thing about, well, what other interventions might we take a look at? That's psychotherapy alone. But then we know that if we add Prozac to psychotherapy, there's a lot of research showing, wow, Prozac's one thing, psychotherapy is one thing. Together, augmented psychotherapy, we see a bigger change. Well, what about that within the lifespan? And now we see the example of MDMA-assisted psychotherapy. So you have these psychedelics that Terence McKenna called deconditioning agents. So now we use the same psychotherapy that's shifting people's epigenetics, but we augment it with this deconditioning agent. And then you see these transformative healings. Similarly, I saw that kind of thing in Peru, and I've seen it in ayahuasca curanderismo, as we call it, within the context of la dieta, of a healing context or a prolonged intervention. You can see the same kind of shifts occasionally. So in summary, as I told you at the beginning, trauma is a big deal in medicine and healthcare. I'm going to get back onto I'm going to come back to the thing. I don't know who's chatting. I haven't paid attention. Actually, there's more. I got to stay with it. Trauma, as I said, is a big deal. We got the adverse childhood experiences. We have PTSD. We know there's treatment-resistant trauma out there. We know people need more understanding and hope. Those segments of the population that aren't getting better with eating disorders or chronic PTSD or whatever it is. A lot of other illnesses described: anxiety, depression, addiction. So epigenetics is opening up a pathway for us to have more understanding about what is it that happened with these. Is it inherited? Is it from their childhood? Is it from their infancy? Is it in utero? Was there an imprint that happened? And is there hope that we can do something about that? And then we have these transformative healing experiences like MDMA-assisted psychotherapy that is providing further hope, because these people, they weren't supposed to get better. They said no one knows how to help them. And now they're shifting from something that it looked so biologically entrenched, so stable. So as we understand the biological underpinnings of that trauma and then the related healing of that trauma, we strengthen the case for that hope for these people. That's why it's so important for these people and for the psychedelic assisted therapy. So we want to make a bigger case of, hey, this is important. These therapies should be made available. These therapies should be covered by insurance, et cetera, et cetera. And so, well, guess what? Because some of them may be shifting epigenetics, and that's what some of these people need. And treatments that don't reach that level are not going to be that helpful to these people. So what's the update on the study? Well, we've got the analysis ready and waiting, as I said, on the first 23. Posts are pending on 10 more. The MAPS trial centers have, we asked them, the ones that we move into the next phase of the study, there's different trial centers all over the country. We want them to help us. Please encourage your participants to participate, and offer saliva samples so we can try to see what's going on epigenetically. MAPS has invited Modern Spirit to continue analysis with USC, basically. I put Modern Spirit, that's wrong. It's all of us, USC, TGen, et cetera, all the team. Analysis in the next phase of the study. So we have this opportunity to increase the power of the study. But we need your support. We need the support to go forward. The next phase of the study is more expensive. It's more lab work. So please, we've got a crowd fund. We're trying to get people to take an interest. We know it's kind of a complex issue. Sometimes people don't appreciate what we're trying to do, but I hope I was able to relay it a little bit for you to see how important this could be and how it is. And there's the crowdfund. Kay will put it in the thing. So I'm going to stop screen sharing, and we're going to switch to question and answer. I want to introduce if Raheel Khan and Candice Lewis are here. Let me try to get out of screen share. How could I get out of screen share? Any moment it's going to happen. Did you get me out?
You are out.
Okay.
Wonderful. And I know that our tech is working on getting, Michael's working on getting, we have Candice on, and welcome, Candice.
Hi.
And we'll be getting Raheel on as well. So Candice, perhaps while we're waiting for Michael to get Raheel on, did you want to maybe give a bit of an introduction? Just who you are, what you're doing, just so our lovely crowd here can know who you are.
Sure. I got on a little bit late, and I got on in the middle of Joe introducing me, so I'm not sure what was already said about me. But my name's Dr. Canis Lewis. I started my research in animal models, looking at how early life stress affects the epigenetics of brain reward regions and how that change in epigenetics and reward regions changes the way that animals respond to drugs of abuse and their behavior in taking those drugs. From there, I got really interested in kind of the inverse of trauma, that if early life stress is so salient and powerful that it has the ability to change brain tissue at the molecular level in a way that shapes behavior in adulthood, then what can the inverse of trauma be? And that's really what led me into the psychedelic research world. And that's when I went and worked with Dr. Franz Woldenmeyer in Zurich. So now I ride both horses. I use the identical twin model design to look at how environmental influences in childhood can shape epigenetics. And the reason the twin model is so important is because we know that genetics are also shaping epigenetics. It's really complicated. But the identical twin model controls for that genetic influence. So it's really isolating the environment. It's really showing that this environmental effect is what's shaping biology and brain function, structure, and behavior. So on one arm of my study, I look at early environmental influences on epigenetics, a brain of gene systems that are involved in mental health. And then on the other hand, I'm interested in the psychedelic world and how can these salient experiences, these healing experiences from psychedelics, could they possibly also be changing us at the epigenetic level in a way that then downstream changes brain structure, function, and behavior? So that's my research world in a nutshell.
Wonderful. Thank you so much for sharing that. And it looks like we also have Raheel on now with us. Welcome, Raheel. And we're kind of just going around and giving a little bit of an introduction. So if you wanted to share a few words about yourself, that would be wonderful.
Sure. Yeah, I'm Raheel Khan. I'm a psychiatrist and a neuroscientist at USC in Los Angeles. And I got involved in work pretty early on in my scientific career, the kind of PhD training. Joe may have mentioned it, but he and I went to medical school together in San Diego, starting back in 1999. And so as a grad student, I got into this area of research generally, and my interest has always been focused on the other reaches of human consciousness, for the evolution of human consciousness in particular. And so I was really interested in looking at the neural correlates to transformations of consciousness, especially with the experience of the self kind of transcending from the kind of personal self, conventional self, to more of the world-centric self that is described in spiritual literature going back millennia. And, intuitively having the sense that if the scientific modern world is to ever have a chance of changing at that fundamental level, so that that's where humanity is living from, we're going to have to understand at a scientific level as well. So started some studies looking at long-term meditators and the effects of psilocybin on brain activity and states of consciousness and self-experience. And actually, Candice and I have in common. I went with one fighter in Switzerland back, I think, from 03 to 05. I was there doing the work with psilocybin and meditation using EEG, my kind of area of expertise. And, then finished up medical school, residency, and postdoctoral fellowship. As a faculty at USC, I've been involved in continuing work looking at meditative practices, mindfulness practices in relationship to depression, addiction, general growth and development, with a continued focus on the relationship between self-experience, or the transformation of self-experience, or relationship, I think, process of who we are, and the way in which that can be shifted to complementary, integrative approach, and that can be more spiritual world, but have more knowledge that is, somehow irrelevant to well-being, and then have long had to get into my interest working with psychedelics, and that's kind of how I came to be involved with the project when I first exposed for the many years. And as he was seeing that he had the opportunity to actually fund a project of this nature, looking at epigenetic relationship healing through the MDMA therapy project of maps, that it would make sense as a preliminary step, or kind of maybe secondarily down the road looking at the changes related to ayahuasca-related healing. I thought that was a great idea, and so I signed on to do what I could to facilitate that reality. Luckily, we met Dr. Lewis about a year later, who actually had the scientific chops to do the laboratory analysis. I have a background in molecular, my area of kind of active expertise of the scientific methodology level is neuroimaging. And so in the long run, I think that's one of the things this project is aiming towards, just kind of the same way that Dr. Lewis kind of wrapped up her discussion of that interest in the full spectrum of how these kind of transformations of consciousness that are related to spiritual healing, how they resonate through the human kind of biofield into all the way from the molecular biochemical level through the neural circuitry and kind of brain patterning reformulation. Certainly, the one thing that has become clear, and I think Dr. Tafur already mentioned that. I saw some of the slides that presented. This commonality of things that psychedelics have the potential towards producing both short and long-term enhancement of plasticity, or kind of allowing the biological system to go into a state of adaptive shift, change, and that can both be seen at kind of molecular patterns and epigenetics, but also at the level of neural circuit rearrangement and opening up the opportunity for forming of new patterns that can take a widely new direction from some of the habituated patterns that are involved. So, in any case, that full kind of molecular brain, body, spirit kind of back and forth communication of change is something that I think there's a lot of hope for the future of kind of medicine and our understanding of how to live, help each other heal from the world we live in.
Mm-hmm. Absolutely. Well, thank you for sharing that, and thank you for being here with us today, Raheel and Candice, as well as Dr. Tafur. So we have quite a few questions here from our crowd. Did you guys want to share anything else about your study or what you're doing before we hop into the questions, or do you want to go?
Yeah, we're going to lose time.
Okay, perfect. So the way that we're working the questions is we're going with the highest voted first. So I'll read those off the Crowdcast for you, and then just let me know who wants to take a shot at answering it here. So the first one we have is from Eric, and Eric asks, would you explain in as simple terms as possible how trauma creates epigenetic changes? So who wants to give that one a shot?
Want to try, Candice?
Yeah, I'll take that one. That's a really good question, and it's not quite understood. One hypothesized model, so cortisol, when bound to its glucocorticoid receptor, inside of this, when cortisol is bound to its glucocorticoid receptor within the cell, it acts as a transcription factor for hundreds of genes. So one possibility is that in response to chronic stress or in response to traumatic experiences, that could be turning on and off certain genes. But that would just be one simplistic explanation. How the overall system is responding to these psychosocial stressors is really unknown. We know the molecular machinery that adds and removes methyl marks on DNA, but how exactly that machinery is responding to trauma is unknown. In one of the papers that Joe cited of mine, I hypothesize about this. I discussed the possibility of this cortisol-bound receptor regulating genes, but that's a big unknown in the field right now. In its it's such a new field. Like, we've really only seen epigenetic marks, which we've known about epigenetic regulation of gene expression since the 40s. But this idea that it's responding to social environment is really quite new, about 10 years old. Well, you were going to pick your questions, but I saw another question that was kind of related. I could just quickly answer. Someone asked, I saw how someone said epigenetics can turn on and off genes. That is correct. So in epigenetic markers can either, and a lot of people talk about it like it's this on-off switch, but biology doesn't tend to work like that. It's more of like I like to think about it as like a mood lighting, like higher expression or lower expression. And DNA methylation is only one of the epigenetic marks. It's a really complex system with a lot more things going on. So genes can be regulated higher or lower in many different ways. Third question that was related was that what's the epigenetic response to effective treatment in PTSD? And the answer to that is it depends. Some genes we see higher methylation and reduced expression. With some genes, we see lower methylation and higher expression. So depending on the gene, it's going to go up or down. And depending on the gene, that will have either up or down effects on, say, the HPA axis, as Joe explained. For example, the common phenotype in PTSD for the HPA axis is a hypo response, and he showed that in the curve. With PTSD, they tend to have lower amounts of circulating cortisol throughout the day. Whereas with depression, you can find either or, but you're more likely to find a hyperactive cortisol response, where people with depression tend to have higher levels of cortisol in response to stress, at least. So it's people like to think in very linear ways, and people like things to make a lot of sense. Everything goes up or everything goes down, but unfortunately, neuroscience isn't that easy. Okay, now you can go to the next question.
I will just add, I just want to add one little comment is like, because what she's saying, it's a mystery right at that point. Like you get it down to that point where you say, okay, this psychosocial interaction, and then you see this molecular response. And so that is a real mystery, but we are alive. So then, the shamans and the people that I work with, well, they don't really, have a hard time with that, because we see that, I see the psychotherapy is affecting this molecular stuff, state of consciousness, like meditation, state of consciousness is affecting it. And then we see, I would go as far to say the love or the lack of love, emotional availability, or harsh parenting or not harsh parenting. And so we're really like, to me, and this is where we get out of bounds a little bit, but it is a metaphysical process that we're observing. In other words, we're not limiting ourselves just because we don't understand the mechanism. We do know what it's like to be alive. We do know what it's like to receive harsh parenting. We do know what it's like to have our, state of consciousness altered. And the reality is, our molecules respond to that. And so we don't know how exactly, but we know that it does. And it involves kind of a holistic, like higher dimensional understanding of like what I would say being alive, from the shamanic perspective. I don't know if Raheel has anything to add, or want to move to the next question.
I think you guys have covered the base as well. At some level, it's a deep mystery, and there are some kind of some mechanistic understandings that we think are probably at some point play a role. I think maybe it's a little bit of a overly detailed kind of aspect of it, but just to point at one kind of mystery is, some people will say, oh, well, maybe the change is happening, from changes at the molecular level. Some neurotransmitter changes obviously seem to get involved. If you're taking a psychoactive substance into the brain, get in the synapses, start changing the way the receptors are responding. And then some molecular changes happen. And of course, these substances do have effects at body, tissues other than the brain, but it seems like a lot of the effects might be related to the changes in neural activity. But the question of whether, for example, that translating into longer-term changes of, inflammatory processes, which, clinically people are seeing really clearly with some of these forms of healing, that is happening because of some molecular change that has, maybe some brain change associated with it, but that's not the key, versus the brain change really might be key to kind of unlocking some downstream molecular changes. And we just don't know. These are the kind of details of how the biology responds to these kinds of healing experiences that are, just starting to be looked at.
Thank you so much. So we have a question here from Lisa, and she's asking if you're measuring changes by evaluating telomere length.
No, not at this time. I have looked at telomere length in association with other psychiatric disorders before, but at this point in time, we're just looking at DNA methylation.
Okay, perfect. Thank you.
I could say that we did think about that kind of thing. And there is, if we get enough samples, which I think there's a good chance we will once we get the second batch to add to the first batch, there are some other methylation patterns that are shown to be associated with molecular age or cellular age that are a separate signal from telomere length. And we were thinking if we get enough samples that we could at least interrogate that way. And of course, if we had enough money, we could also look at telomeres.
Money.
The money.
A great idea. Please help us, and we'll do it. We'll try.
Yeah, exactly.
Yeah. Fortunately, with the method that we're using to look at DNA methylation, as Raheel was saying, once we have enough samples, we would be able to look at an epigenetic clock.
Okay, perfect. And it's all about the money, and the link is right in the chat, the GoFundMe. Eric also commented, he said, I think there's something mystical about how Candice used the mood lighting metaphor and then her lighting dimmed.
Yeah, explain the mechanism of that. Maybe it was just your computer, Eric. Maybe it only happened to you. I don't know. I didn't see it.
We have a question here from Brendan. How well do epigenetic changes as measured in blood correlate to clinically relevant epigenetic changes in neurons?
I'll take this. So this is like we lovingly refer to in epigenetics as the tissue issue. So as you're pointing out, we're discussing changes in behavior and symptoms in psychiatric disorder, which the tissue of interest would be brain. But we're not looking at epigenetic changes in brain. We're looking at epigenetic changes in peripheral samples. So a way to think about this is we're really looking for a marker of change, not the actual change itself. Animal studies have shown correlations between changes in saliva or buccal cells, blood cells, and brain tissue. And also human postmortem studies have shown correlation between brain tissue and peripheral samples. However, we're still not looking at the tissue of interest, and we're not pretending to. So we're not saying we're showing brain changes. We're just showing another sample that may reflect it, another tissue that may reflect it. For example, I watched a talk the other day that this person was interested in epigenetic changes in the sperm line, so in sperm cells. And in animal models, they were able to show that I believe it was blood, that changes in blood reflected changes in sperm. So it wasn't their tissue of tissue of concern, and they won't be the exact changes, but they still are markers of change, if that makes sense.
Joe or Raheel?
Yeah, I think the important part, because there is that question, and that's part of the deal. And I have my theories about the network and how there could be tissue specific stuff that I would assume would be linked up, and there's evidence that there is some linking of methylation pattern, imprinting in different tissue areas. And then, what we're also tracking is in the areas that we are measuring, are we seeing a change in the response to the intervention, to the therapy? So we're actually comparing, where were they before to where they are now. So we're looking at them as a window of our first question is the clinically relevant question that you're bringing up is, yeah, that's real. We want to know that. But first, do we see any epigenetic changes that we can measure and report on in response to this psychedelic assisted therapy? So that's, with our model, that's the first step. And then we start correlating and exploring beyond that.
Mm-hmm.
One other thing I would say, just add to that, is it could very well be the case that some of the kind of more neurally specific genes that we're looking at, like for example, the 5-HT2A receptor, and or the some of the neurotrophic growth factor, BDNF, might be changed in the brain, but not in the cells we get from the saliva, which is basically the epithelial cells of the lining of the cheek, combined with some of the white blood cells. It's those two populations that we're really looking at when we look at salivary DNA. And so, you know, yes or no, maybe the same changes to the genes in the peripheral, you know, rest of the body are changing. You know, those epigenetic modulators are happening in concert in both tissues at the same time, and maybe they're not, and we won't know that because we're not going to be taking people's brain samples. Like Joe said, you know, if the correlation is there, it's still of interest, and we do know, as Candice was saying, that, you know, looking really systematically across brain and these other tissues, that there are some correlations. It's not a one. The correlation is not 100%, but it's, you know, it's not zero either. But the only thing I wanted to add is just that it could also be, you know, one of the things we're looking at is some inflammatory gene, and it could be that maybe changes in the inflammatory genes are not even happening in the brain, that those are actually happening peripherally, and so we wouldn't catch it even if we were looking at brain cells. So, you know, there may be some advantage to us looking in the periphery for at least those particular genes that we're interested in looking at.
And then ultimately, you know, the real impact of this study and the power of this study, that really is like kind of like the big deal, is we're piggybacking on the MAPS trial, which is collecting all this psychological stuff where they're actually going to, the clinical correlation is taken care of. You know, that's not a mystery. Our team is just piggybacking, tracking. We're going to have a lot of clinical correlation information from what they've been reporting and publishing all this time, and now correlate that directly to whatever change we do observe. So we are going to get something clinically relevant.
That is so exciting and so good for the future of medicine. We have a question here, Joe. This is on your slide share. Can you share your reference list that you went through in the slides? Is that possible?
Yeah, I guess it is, but I'd have to like put it together. I mean, of the slides, is this recording? This is like part of the crowdcast, and they just like flip through it?
Yeah, they can go back. We also do send out a summary email Wednesday, Thursday-ish with some links and stuff, so we can put any links in there.
I'll do that. I'll get those together for you, Shannon, and you can put those in there.
Okay, perfect. Thank you. So the next question here is from Kate. And Kate asks, what is available for atheists, people who identify as non-religious and non-spiritual?
What is available? The same as everybody else? That's my first answer. I don't know.
Hard to know what she's asking exactly by available.
I presume, and please, the commenter, correct me if I'm wrong. What I'm hearing is perhaps for an atheist or a non-spiritual person, they might be a little bit off-put by the amount of spiritual language used around the psychedelic healing experience, and think that maybe it's not made for them. Joe, I'll let you take on that.
Yeah, I'll just like comment a little bit. So this is, you know, of course, I'm talking all spiritual, and I'm on that train. But you know, the way that those things are merging, and like, how does the secular world or the atheist world talk about these things? So the first is like, you know, Johns Hopkins research, and Raheel can help here. But it's like, you know, they have this mystical scale, the mystical experience scale. So this is a secular. You don't have to believe in anything. The questions are, did you experience like like space and time? Did you get lost there? Did you have a different kind of experience with space and time? Did you experience oceanic boundlessness? You know, did you experience some kind of gnosis, like some kind of knowing that exists beyond your consciousness? So that's the way, and Raheel can speak to this a lot more because mindfulness research is all about this, you know. How do we talk about things in secular terms and we're kind of like in that blurred area where some people think about these areas very spiritual, and other people are like, well, let's just talk about it in terms that everyone can relate to. So I don't know, maybe I'll ask Raheel.
Yeah, I mean, I think that the question of language when it comes to talking about these kinds of experiences is really fundamental. And you know, one thing I can say is that you know, when I use the term spiritual, I actually am not referring to any sense of knowledge about or belief in a particular philosophical or systematic understanding of like the relationship between something like the divine or God and the rest of nature. What I'm actually referring to could easily just be a substitute term of consciousness, the quality of awareness. And so I think what it points to, that term, is something about the experience, which is really something that can be accessed just through your knowing and no other way, of something beyond the conventional kind of separate self experience. And so, you know, it gets into this area it just really takes some time to unpack how to use language in a way that isn't loaded for people who really have a pretty clear sense. Like, you know, atheism, of course, is different from agnosticism. You know, there's a kind of active disbelief in a kind of supernatural power of some kind, or God. But really, when it comes down to it, at the level of kind of clinical relevance of you know, these concepts, spiritual healing, you know, that term spiritual healing, really what that is pointing to is the experience of some kind of change of consciousness, where you don't feel so isolated and separate from what's outside the physical body. And usually there's an emotional kind of intensity that we use the word like love to describe. But again, you know, these are all just words that's the best we can do, is to try to communicate words. In fact, we're trying to point to experiences that are profound and meaningful, and change our sense of, you know, the relationship between the inner and the outer, between the self and the other. So I think it is possible, just by kind of recognizing that these words are actually not meant to portray some worldview as much as to point to something about experience and the power of experiencing, you know, altered states of consciousness that are altered from what? Altered from the conventional state, where we seem to be these separate kind of billiard ball, kind of Newtonian physics kind of analogy, you know, not actually part of each other, but separate from each other, and, you know, in competition with each other. And it's just a very different realm of experience that we have access to through these kinds of technologies, whether they be meditative practices that can be done whether you believe in God or not. A lot of the traditions, actually, like Buddhism, actively don't have a conception of God, but those practices are central to these, you know, traditions that are even older than those organized religions of the shamanic practices that use these substances that are now coming to be rediscovered by psychiatry.
I just want to, just because that happened, you know, I was reading an Albert Hoffman quote, you know, that's why. So then it's back, and I'm sure we should switch it into Swiss. Should we switch to Swiss? Now. But Albert Hoffman, I was reading a quote from him, and he was using that language, like Raheel says. I wasn't trying to trigger or exclude any kind of, you know, atheist. It was just saying, this is what Albert Hoffman said, and it was interesting to me because he was talking about shamans and shamanic culture. So I just want to read a little bit more of his quotes to flesh out his language. This is not mine. I was just reading from him. A very deep problem of our time is that we no longer have a spiritual basis in our lives, says Albert Hoffman. The churches are no longer convincing with their dogma, yet people need a deep spiritual foundation for their lives. In the past, this foundation was built on accepted religious creeds, which people believed in. But today, such beliefs have less power. We cannot believe things that we know are not possible, that are not real. We must go on the basis of that which we know, that which everybody can experience. On this basis, one must find the entrance to the spiritual world. Because many young people are looking for meaningful experiences, they are looking for this thing, which is the opposite of the material world. Not all young people are looking for money and power. Some are looking for happiness and satisfaction, born of the spiritual world. They are looking, but there are very few credible sanctioned paths. And of course, one path that still delivers for young people is that of the psychedelic drugs. So his concept, his language was talking about happiness and satisfaction, those kind of concepts that some people would call that spiritual language. You know, how do you talk about that stuff? We're not talking about just molecules, genuine holistic concepts like happiness and satisfaction.
So I would quickly add to wrap it up with all of that completeness. I validate the commenter and anyone else who feels adverseness to spiritual language. There is so much because of the dogma and the actions of mainline religion over the centuries. There are so many people carrying spiritual trauma, and it might be very hard to wrap your mind around a system that uses this language. So I acknowledge that it can be adverse for people. And I want to offer to you a solution that when you hear words that don't sit right with you, for example, I have really struggled with the patriarchal nature of religion and how Lord and male pronouns are used almost exclusively. So replace it with words that work for you. If someone says God, replace it with universe. If someone talks about a spiritual connection, just replace it with connection. And really, one of the ways that we could boil all of this down about the psychedelic experience is that it facilitates connection both with self and with the outer world. So validate that it can be really hard with this language for so many reasons, and offering a tip to try to replace it. I have a TED Talk you guys can all check out where I really talk about connection being one of the ways we can boil this all down. But I also love the discussion about language. It's just limiting. It is. Okay.
Can you put the link up to your TED Talk in the chat? Did you?
Could do that.
Please. Well, thank you all. Those are all amazing answers to a very interesting question. So thank you so much for that. We have a few minutes left, and we will answer a few other questions if that's okay. We have one here from Amy. Is there a nutrigenomic approach to
medical practice? And
It started. TED Talk started. Let's switch to the TED Talk.
No worries. So Amy asks if there's a nutrigenomic approach to reduce methylation tendencies.
There is. Functional medicine is definitely interested in epigenetic modification through nutrition and nutrigenomics and herbal medicine and pharmaceuticals. So I'm not an expert in that, but I know that that's definitely a focus of in functional medicine. You can learn a lot more about that approach. That's my quick comment. Hand it over to the team.
I'm not aware. Candice is probably the most likely of the three of us to be aware of, actual real studies that have gone on looking at nutritional approaches to changing epigenetic patterns. I'm not aware of any, and my sense is, because this is still such a new field, and we're still at the point where, for most psychological symptoms or either psychiatric or clinical diseases of another kind, cancer, what have you, we don't have really strong associations between the altered epigenetic state of a given gene or set of genes and that particular pathology, you could say. So because we don't have that, it's hard to then give a nutrient of some kind and see, like, okay, does that that problem that we know is this association between that genetic state and that clinical problem, that we're now seeing that's benefited. And so I think that the science is probably just too young to have those kinds of studies be meaningful yet, but I don't know if you know of any, Candice.
Yeah, I can't say anything right off the top of my head because nutrition is not my realm, but for sure there's a large body of literature looking at nutrition and diet and how that affects epigenetics. I would say it's primarily probably still in the cancer realm. Epigenetics and environmental exposure and cancer research has been going on much longer than exposures, epigenetics, and mental health.
Right.
One thing to remember always about food science is it's just really hard to conduct. So it's, to be really isolated and studying the true effects of specific diets, one would have to have people living in a lab for a long period of time. That's not a thing. So you heavily rely on self-report, which is notoriously hard for everything, let alone what we're eating, right? So that's the thing. Nutrition science is hard, but for sure there is large body of literature looking at. So one way that's just done a lot is just epidemiologically. So looking at this region tends to eat this way, right? The Mediterranean diet versus this region tends to eat this way, and kind of comparing across that epigenetically and disease-wise. I'm sorry I'm so dark. I can't do anything about it.
No problem. Does anyone else want to comment on that before we go to the next question?
I think they, that's the scientific perspective. They've covered it. I think it's fair.
Perfect. The next question here is from Chelsea. She says, Joe, can you comment on the jealous nature of ayahuasca? Oh, where did the question go? There it is.
Ayahuasca. Are you thinking about Raheel, Candice? Raheel, Candice? It's kind of like a little separate from what we're talking about. I'll just open with that. But just to comment briefly is that's something that people say down there in the Amazon. Some people say, and what they mean is that the people shamans that I work with ayahuasca. They say once you start working with ayahuasca, the ayahuasca wants you to stay with her and not drift over to other substances. So I don't know, is that at me? Because now I'm working with MDMA research, and now I'm is ayahuasca angry with me about this? Like I said, ayahuasca told me to do this. So that's where I'm at with it. And so I'm at peace with ayahuasca and any jealousy around that in this moment. So thanks for your question.
She also actually added something in the bottom of it. And she says, do you see MDMA psilocybin research as a means to an end, in brackets, getting more ayahuasca research approved, or as an end in itself?
I think that that's all going together. And so there is ayahuasca research. A lot of the ayahuasca research is happening in Brazil. Currently just getting, right now they had to struggle to get the MDMA approval with the DEA exemptions that they have to do research with psilocybin and MDMA in the United States. I don't know of any ayahuasca research exemptions within the United States currently. So that really limits how much research is being done. There are groups that want to study ayahuasca that way, but meanwhile, South America has open access and they are doing research on ayahuasca. So it is happening. And so, I think that it's all flowing together, and really the spirit of it, as some people, I can say for myself, that's really kind of live very closely with that tradition and these plants, and that the most important thing in my perspective is helping people. And that's why I got so deeply involved with ayahuasca and plant medicine. It wasn't because I want only people to do ayahuasca and not MDMA. If I'm up here and I see somebody that is going to be able to benefit from something, I want to help them find that. And I believe that's what the plants want, is my, getting on the shamanic, wild and weird spiritual world. That's my belief. And so I don't really see a big issue there. But I do, that's one of my messages is like, well, what can we learn from the traditional cultures? And that's what I opened up with what Albert Hoffman was saying about how to use this stuff and what goes on. When we were just talking about the mystery of the epigenetic change and at the molecular level, is what goes on in those sessions. So, me as somebody that works with ayahuasca that way in ceremony, I honor the therapists of the MDMA trial because I know the art of what they're doing plays a huge role in what's going on for people. And we can see that in the placebo arm of the trial, without the MDMA, that it's a big deal. And then with the MDMA, still how they work with it facilitates what goes on there. in this mysterious world where epigenetics may be shifting, where molecules may be shifting. So the art of healing is a part of it. So, I always want to honor that part of it, and that's my interest. And I want to honor MAPS for doing that. It's MDMA assisted psychotherapy, and the training that they do is about helping therapists help people find, results with whatever their problem could be.
Absolutely. So in regards to the therapy, post-successful PTSD therapy, what changes are exhibited in the epigenetic alterations of affected genes?
That was one that I attempted to kind of get at in my first long answer, just that depending on the gene and even depending on the CPG methylation site within the gene, we either see an increase or decrease of methylation, which then leads to an increase or decrease of expression.
If I understood the question, it's what changes are observed in successful treatment of PTSD or successful treatment of PTSD through MDMA assisted therapy.
Just
post-successful PTSD. What
PTSD
epigenetic alterations?
In general, yeah,
psychotherapy or whatever it is.
Yeah, the only thing I would add to that is the studies are not all they do not all show the same thing. So some studies have found some association, with the two things that have been most frequently observed to be altered in PTSD, as mentioned, were the glucocorticoid receptor and its kind of molecular chaperone partner protein called the FKBP5. And so some studies have shown, this altered methylation of the glucocorticoid receptor. But there are different sites, as Candice is pointing out, different sites. And in general, in addition to different genes having a different relationship between methylation status and expression, there's also, is the methylation in the promoter region or in the non-promoter region that, affects whether it seems to be associated with increases or decreases of the expression of the gene. So a complicated answer, I think the bottom line is we don't yet know. We don't yet know. And we certainly don't yet know about the pattern that may be specific to the using an MDMA assisted therapy approach. We're the first folks to try it.
And it's great work that you're doing. So is funding the main barrier to holding back your research?
Now it is, because now it's just, first was access to the participants, so we needed to have broader access. So, MAPS is limited in their negotiations with the FDA. When are they going to be able to do this, with how many people, and, when's that going to happen? And, do they want us to be the ones evaluating that? And so far in the next phase, they've said, yes, we'd like you to try, to go. So we're going to open the access. So then that involves participation of all those different trial centers to say, hey, to their clients, hey, try this, because now we're going to do in the COVID era, it's going to be communicating with them virtually and, through mail, like ancestry.com kind of thing, and swabbing, and maybe swabbing on Zoom. And so all those steps have to happen to get the samples, so we need the team, everybody involved, and the participants to consider, being part of this. So that's important. And then we need the money to continue to pay for the coordination of the sampling, and then ultimately the analysis of the sampling. So the money ultimately is, once we have the money, it's a lot easier to try to make the other stuff happen, but we're doing both together.
Yeah, I will throw in that money is a huge issue because genomics research is so expensive. With that, I'm going to run. I know we're rounding up, but I have hungry babies. So thank you all. I love being on the team. Keep up the good work, and bye, everyone.
Thanks so much, Candice, for joining us. So we are coming to time here. Is there anything, Raheel or Joe, that you wanted to add or share with the group we have here in Crowdcast as a bit of a closing?
Well, you want to say something? Then I might have something to say also. You okay?
It was a pleasure to join in for a bit. Thanks for the invitation, Joe, and for funding this project. It's been really fun to be really now in the midst of it, and seeing that we have these opportunities to keep moving forward, and the relationship with MAPS has continued to deepen. So that's been It's been great.
Yeah. No, I just want to say it's just an honor. Thank you for having us. It's amazing. Thanks for giving us this opportunity to share about our project. It just, for me, it was so fun and so fantastic to be part of this trio with, Raheel and Candice to answer these questions. I feel like it's such a broad, interesting team that we have. And, the last part of me talking about like my spiritual journey, and again, with all due respect, as Candice said, for everyone's experience, but I was just telling about me. Like, this is how I came to this. So that's what I was sharing. And so for me, it's part of my journey. And so I'm sharing just from my experience, that is interesting. It's very personal and got me inspired to try to push for this. And so we're really happy and proud that we made it this far. We look forward to doing more, and we've got our results coming down the road, and we'll see. Maybe we won't have that much to say. Maybe we will. We're excited. We're excited, but we the more numbers that we get, the more participants, the more we're going to be able to say. So we're excited to keep the study going. We're excited to have people help support the study, and I hope that people understand. If they have more questions, contact us through Modern Spirit or through my website, drjoetafur.com. And if they want to understand more about how to support or what they didn't get from these talks, that's what we did today.
Mm-hmm. And people can always come back and watch the talk because they are recorded. So just remember, you can log into your Crowdcast, and then I believe we'll send out that list of resources that you were talking about in your slide in the follow-up email that we send out in the next couple of days. And we look forward and are excited to see what you find in the studies. All very important work, and so grateful that you're here to join us today to share that with us. This is definitely needed in medicine, and can definitely help a lot of people going forward. So thank you so much for being here to share that with us.
Thank you.
Thank you.
All right, so just to wrap up the episode today, I would like to take this time to thank our speaker for the day, Dr. Joe Tafur, and his colleagues, Raheel and Candice, for joining us. Thank you to all of you for being here to support this series, these speakers, and this important research. Remember, you can support MAPS research by going to mapscanada.org. And of course, you can definitely help support the research, very important research, with Dr. Joe Tafur at the link that was provided in the chat there. Also, thank you to our episode sponsors for the series so far: Enhanced Therapy Institute, Spores Lab, Stilopedia. And if you're interested in sponsoring an episode, you can contact sponsorship@mapscanada.org. So our next episode will take place next week, Tuesday, 5 p.m., October 27th, with 5 amazing visionary artists. We have Chris Dyer back with us. Jake Cobran will be joining us from, I believe he's in Bali. Amanda Sage, Chor Boogie, and Simon Heidick will also be joining us for our Visionary Artist panel. So this would be a very interesting episode. You don't want to miss it. And you can check out the episode on our website, mapscanada.org/webinar. And that is it for this week. So thank you so much again, Joe, for joining us and sharing that important information, and to your colleagues as well. And thank you everyone for tuning in today. So have a great week, and we will see you next Tuesday at 5 o'clock p.m. Bye for now.